In this lab activity, we're going to learn about osmosis and diffusion. So we're trying to set up a greenhouse, and there are 6 subterranean water sources, but which one would be the best one to tap into for our greenhouse? There are varying amounts of dissolved materials within each of them. In this lab activity, we are going to compare them to each other and determine which is the best water source for our greenhouse. Some information we need to know to solve this. First off, plants need to be watered with a hypotonic solution compared to their own internal cytoplasm. When comparing solutions, a hypertonic solution has more dissolved solutes than another solution, whereas a hypotonic solution has less dissolved solutes than another solution. If 2 solutions are equal, we would say that they are isotonic. An isotonic solution has an equal solute concentration compared to another solution. The roots of a plant rely on diffusion and osmosis to draw in water from the soil. Diffusion is a property of liquids and gases. Diffusion means the movement of molecules, and molecules in liquids and gases will attempt to move to equalize their concentration gradients. Diffusion wants all solutions to be isotonic. Both solute and solvent can move to equalize concentration gradients. These are the 2 major components of a solution: the solute, which is the dissolved material, and the solvent, which is the dissolving material. In diffusion, molecules will always move from where they are in a high concentration towards where they are in a low concentration. Molecules will move to equalize concentration gradients. Osmosis is specifically the diffusion of the solvent or liquid part of the solution. When a solution is hypertonic, the solute concentration is higher compared to another solution, but the solvent concentration will be lower than that other solution. Diffusion is movement from high concentration towards low concentration, and so even though the solute will try to move out of a hypertonic solution, the solvent will attempt to diffuse into that hypertonic solution. You can think of this as the solvent trying to dilute the solution with the higher solute concentration, and this is osmosis. Osmosis is that diffusion of the solvent. Selectively permeable membranes and barriers allow certain molecules to pass through, but they block others. The dialysis tubing used in this experiment is just such a membrane. So are the plasma membranes of our cells and also of plant cells. The solutes that are in our solution will not be able to pass through, but the solvent, the water, will be able to move. So how are we going to tell which water source is best for our plants? Well, we will run a test where we compare each of our samples against a solution that is similar to what we would find inside of a plant cell. If the samples gain water, it means that they are hypertonic to the plant cell's internal fluids and would not be good for our greenhouse. However, if the samples lose water, it means that they are hypotonic compared to the plant cell's internal fluids, and that would be a good water source. Watering plants with a hypertonic solution would dehydrate and kill them. It would actually take water away from the plants, which is what we want to make sure the solution we're watering our plants with is hypotonic compared to its cytoplasm. So step 1, we're going to take a strip of dialysis tubing and we're going to tie a knot into one end of it, making a sealed pouch. Step 2, we're going to measure 25 mils of our sample of interest. And so we have 6 different samples. We'll start with sample A, measure out 25 mils of that sample. We are then going to pour those 25 mils from our graduated cylinder into the dialysis tubing, making a little pouch. We now want to tie a knot on the other end of that dialysis tubing bag, making a sealed container, a sealed pouch with our solution of interest. We then want to get a mass measurement of that dialysis tubing pouch. We want to get a mass measurement of the solution inside. We're going to record this data in our lab report. We're going to repeat this process for the other 5 solutions as well. We have 6 total solutions. We should end up having 6 pouches with each of those solutions, and we should record a mass measurement for each. We should also be careful to make sure we know which solutions are in which bags. After recording the initial mass for all 6 solutions, we are going to prepare our comparison solution. So we now have 6 cups containing a solution that matches or is similar to what the cytoplasm within a plant cell would be like. We are then going to drop our strips of dialysis tubing bags into each of these cups, and for 30 minutes, we're going to let all of them sit. At the end of 30 minutes, we're going to take those dialysis tube pouches out of the solution they were in and get what's called the final mass measurement. And we're going to do this for all 6 samples, recording individually how much the mass of that dialysis tube pouch was after a 30-minute incubation. We're then going to calculate a few values for each of our solutions. First, we want to figure out what the change in mass was. And we calculate the change in mass by having our final mass minus our initial mass. Now, it is possible for this number to be negative if our final mass was smaller than the initial mass. We also want to calculate a value known as the percent change in mass. And to figure out the percent change in mass, you take that value we just calculated, the change in mass, divide it by the initial mass for that particular sample, and then multiply that number by 100. Again, it is possible for this number to be negative if your change in mass was negative. You will have a set of data from your run of this experiment. Repeated measurements have more confidence than a single measurement, so you will compare your values to the average of several other runs of this test. You will end up graphing the percent change in mass for those 2 data sets for all 6 samples. You'll have your measurements and then also the average measurement of a group. And these are tasks that you will accomplish in your lab worksheet. If there are any questions, please do not hesitate to ask.